Okay, welcome to the Bio 101 review video. Today we're going to go through glycolysis, Krebs, and ETC all in one sequence. I think that's important because the images that we usually use are static. So, first things first about glycolysis. This is just happening right in the cytoplasm of the cell. You don't need mitochondria, and you don't need to engage anything else. First things first, you have your glucose. First step is to simply add a phosphate here. That means you use one of your ATPs. Your next step is you're going to add another phosphate here. So that's minus another ATP. So that's minus 2 so far. What's going to happen is that this molecule, fructose 1,6 biphosphate, you don't need to know that name. Biggest thing is that it's super unstable. So it's going to turn into these three little forms, or these two forms. And although this product, and you'll understand more in chemistry with the orientation of molecules really matters. So this product right here, glyceride 3-phosphate, again, you don't need to know the name. This one's gonna proceed, but all the rest of this, what we call DHAP, which is just kind of a small name, this will all just become this glyceraldehyde 3-phosphate. So the big key is that in these first steps where you're investing energy, you're gonna make a lot of this product. Through a series of chemical reactions, you're going to end with the pyruvate that we've talked about. And that is just a 3-carbon molecule. That's it. So in this exchange, you're actually going to gain a couple ATPs because see, you're going to lose these phosphates and they're actually going to recombine with ADPs into ATPs. And since you're doing this not once, but twice, see, because remember, this one is also going to come over here. This happens times 2 for an equal of 4 ATP that you're going to make. So additionally in here, you're actually going to generate one of those key molecules, an NADH as well. So all you really need to know about glycolysis is that we did this entire process to get to this 3-carbon from this 6-carbon. In order to do that, we had to invest 2 ATP, but in the end, we got out 4 ATP and one of these NADH molecules. So net energy-wise, we're actually coming out on top. Yeah, that should be a plus 2 ATP. So net energy-wise, glycolysis is actually a good energetic reaction. You just need to provide the glucose to do it. Okay. So, we started with glucose, we've ended with this pyruvate. The pyruvate itself needs to have one more reaction happen before we transition into Krebs. Biggest thing is that this acetyl-CoA molecule, and remember, this can come from proteins for energy and fats to make this big chemical structure. Oops. So initially what's going to happen is that one of these carbons, this is going to interact right here. One of the carbons actually goes off and becomes CO2. That's a byproduct. During this reaction, one of these NADHs is charged with an electron. So remember, when you add an electron, something is becoming reduced. And what you ultimately combine is this CoA molecule with 2 of these carbons. Because remember, this one went over here to CO2. So your ultimate product is going to be this CoA plus the 2 carbons. So although pyruvate is the key product of glycolysis, this final step, and it's kind of on its own independent, has to combine this CoA with the 3 pyruvate molecule to make sure that you add those 2 carbons right here. Ultimately, this is what we can call acetyl-CoA. And this is our ticket into the Krebs cycle. That's the major connection. So this process is good because it allows proteins and fats to incorporate into the energy process. We expel one of those gaseous CO2s, we breathe that out, remember? We actually do generate one of these charged NADH molecules, and ultimately we generate the compound that allows us to enter the Krebs cycle. Okay, so we're at the Krebs cycle now. Key molecule is that acetyl-CoA, and this is the beginning of it. What initially happens, and remember this is why this is a cycle, is you have the 4 carbon, and this is oxalacetate. I won't ask you the names of these, but it is important to me that you understand where this 4 carbon is coming from, and that it is a cycle because these 2 things combine in the initial step to form a 6 carbon molecule called citrate. Now, there's some chemical differences between citrate, which is 6 carbon, and glucose. So just know that they can't react the same and things like that. So the main thing with the Krebs, which now we are inside the matrix of the mitochondria. So this acetyl-CoA actually has to come from the cytoplasm in the cell. And the first step is that these 2 carbons are going to fuse in with these 4 carbons in this first reaction. In the next couple reactions, what happens is that you actually shed 1 carbon. So see, we're missing 1 here, and it's gonna go off as CO2. Again, just a byproduct. It goes away, it goes right out of the cell. During this step, we charge 1 more NADH. If you're looking for a distinguishing feature of the Krebs cycle, it's that it's going to produce lots of these. And remember that NADH is what is going to be powering the entire electron transport chain. So these molecules are really the key to making ATP. You can get away with some in glycolysis, but not all of it. So we're gonna repeat this process with Krebs. We kick 1 carbon off, the CO2. The electrons from that exchange are taken up by NADH from NAD+. So every time that we charge another NADH, we're charging up that electron transport chain. There's 2 intermediate steps here, one in which we have a GDP plus PI that can become a GTP. This is a lot like ATP and it powers some molecules. So it's a similar idea, it's energy essentially, but you can just treat it like an ATP for the most part for this class. In the second step, we generate that secondary molecule, FADH2. This is also carrying electrons, just like NADH. It's just not as common and it's not as an effective transporter, but it's gonna serve the same purpose. So we have a couple more steps up here that essentially turn this 4 carbon molecule into this conformation of a 4 carbon molecule called oxalacetate, and we can start the cycle again. Now, for the purposes of this class, you're not going to need to know the chemical differences between this molecule and the final oxalacetate. You can just know that there's 4 carbons in this one, and there's 4 here. Within all these steps is an enzyme. In this case, this final step is going to produce 1 more NADH. And that's the key with the Krebs cycle. is during this whole cycle that you're powering, every time you power glycolysis through, you're going to produce 1, 2, 3, 4 of those NADH molecules. That's the key of this cycle. That's the biggest reason that this cycle happens, is to produce these things right here. It's fine to produce GTP. It's fine in glycolysis to produce some ATP, a few NADHs. That's fine. The whole point of the cycle is to add and drop carbons continually, because each time you do, you're going to shed CO2s, but mainly you're going to generate these electron-carrying molecules. So remember, with redox, NAD is being reduced because we're adding this negative electron, right? That's a reduction to NADH. So we have Krebs. It's connected to glycolysis from this acetyl-CoA. We are not directly connected from this process to the ATP or to the electron transport chain. Not directly. Only through these NADH molecules are we actually connected. So for the test, I am going to be writing ETC for electron transport chain. So definitely get used to that. And we are going to be inside the mitochondria on its borders. So essentially, here's the cytoplasm of the cell. Here's the mitochondrial border. Remember, these are just plasma membranes. And here's that intermembrane space. All that we want to do here is transport as much and make a gradient of those H+ ions as we can. Okay. So to begin, let's do these. So like I said in class today, you do not need to know each of the specific electron transporters and carriers. What I want you to know is kind of what we covered in that exam 1. What is active transport and what is the power of a gradient? Okay. Each one of these complexes, its whole role is to shuttle as many H+ ions out of the cell. That's the role of each of these complexes, okay? We want high, this is notation for concentration, we want high H+ concentration here. We want to load all these H+ out here. Now, to form a gradient and to go against the 50-50, we need to load all these out and we need to use energy to do it. NADH donates an electron and becomes NAD+. See that little electron? That's going to power through each of these complexes and power each proton or little hydrogen ion, the little plus. Each time the electron goes through, that's what's powering this through here. That's where the power comes from, is that electron. So eventually the electron is basically used up, but electrons are super dangerous. However, if you pair it with O2, you just produce water and everything's okay. So what we've done is used an electron to power all these active transport pumps, and we're pumping all of this hydrogen, we're pumping it all out, right out there. So now we have all this massive hydrogen out here, not as much in here. So I want to keep that, but All right, so this reaction is still happening because you need to get rid of that electron. And that's what oxygen does, that's why we breathe it. And remember that, oh yeah, so FADH2, this is another electron donator. It does the same thing as NADH is doing to all these. It just goes on this complex. You just need to know that they have the same function. They donate the electron, and that's fine. What happens next is that all these hydrogen ions, they are trying to get back in here. As each of them goes through, this is the only enzyme that you do need to know the name of. This is the ATP synthase. For every hydrogen that goes through, and there's tons of them that are going to go through, remember these little turbines are going to change basically in a rotation like a motor. The first step in the motor is that it loads ADP plus 1-phosphate group. The second step of the motor that's being turned by the binding of each of these, and each of them is going to want to come through, that next motor turn forces that bond together, and it uses the energy of the H gradient. Okay? So that's what's powering this motor, is essentially that all these want to bind so badly. Because every time that one of these binds, one of these steps happens. And the third step is that ATP is released. And then we go back and do it all over again. That's the whole importance of ATP synthase. That's where we force this endergonic bond together. Because remember, it takes energy to form a bond between two things. And it takes a lot of energy to form that third phosphate to become ATP. So that is the whole broad point of the electron transport chain, is that we use NADH to build H plus gradient, H plus gradient fuels this reaction, ADP plus P equals ATP. And ATP can power everything in our cells, all of our complex processes. Now this whole time, everything can only happen if you've got that O2. And remember that the ETC, it's unique because it's not directly connected chemically to the other two processes, the way glycolysis has to make acetyl-CoA to go into the Krebs. The only connection is all of these NADHs. And remember, at this stage you have like 10 NADHs or something from the previous processes. The previous processes are there to produce these molecules. That's on the test. That's the key thing that powers all of this. So each of these pumps being powered by an electron, that's section 1 stuff, but it is really good to know. All right, so that finishes up this video. Email me if you have more questions or clarifications and we can do some more.